Hi, I'm Shiloh, and for my Connections project, I've chosen to make an edible plant cell model. As a visual and hands-on learner, I thought this would be a fun way for me to retain some of the information we've learned throughout the term, specifically plant cell structure. The ingredients I've chosen to use, the base will be made out of Rice Krispie treats. Will be covered in icing and buttercream, and the actual organelles will be made of edible Play-Doh, or some may consider it fondant. Let's see if I can create the image from the book. And there you have it. An edible plant cell. Alright, and here's the final product. So just going over the parts of the plant cell. We have the green portion as the cell wall, which is a layer of cellulose that surrounds the cell. It can be flexible and sometimes rigid. It provides both structural support and protection for cells. And then the yellow portion, yellow gel, is the plasma membrane, which encloses the cytoplasm. It's a semi-permeable membrane that's made up of a phospholipid bilayer. It separates the inside of all cells from the outside environment. Next, we move into the cytoplasm, which is the orange buttercream. The cytoplasm is a thick solution that's enclosed by the plasma membrane, and it's made up mostly of water, salts, and proteins. Then we'll move on to the nucleus, which contains the DNA. On the outside, the indentation here are the nuclear pores, which are protein-lined channels that regulate transportation of molecules between the nucleolus and the cytoplasm. The incisions here are the nuclear envelope, which is a double lipid bilayer that surrounds the nucleolus. Then we move on to the centrosome. It's the organelle where microtubules get organized and it regulates the cell division cycle. Then we'll move to the rough endoplasmic reticulum, or rough ER for short. It has ribosomes attached, which are the red gel. It's part of the endomembrane system where proteins are synthesized. Then we'll move down here, which is the smooth ER, or the smooth endoplasmic reticulum. This is the organelle where lipids and some carbs are synthesized. It also uses enzymes that aid the liver with detoxification. This little guy here is a vesicle. It's a fluid-filled sac that forms during secretion, uptake, and also aids in the transport of materials within the cytoplasm. And then next we have the lysosome. This is the organelle that contains degradative enzymes. It's also enclosed in a membrane. Next we have the Golgi apparatus. It's the processing and packaging center. It modifies lipids and proteins and packages them into vesicles. Next, we have the mitochondria. It's the site of cellular respiration and ATP synthesis. It contains a double membrane, an outer and an inner. It has the cristae, which are these folds here, and then the inside is the matrix. Next, we have chloroplasts, which are also enclosed in a double membrane. The inside is filled with thylakoids. A stack of thylakoids is known as a granum. Chloroplast is where photosynthesis occurs, as well as carbohydrate formation. And then we have the cytoskeleton that is made up of microtubules and filaments. Next we have the cytoskeleton that is made up of microtubules and filaments. And then ribosomes, which are the small red gel dots. Ribosomes are where polypeptides and proteins are formed. And then finally, we have the central vacuole, which is often the largest organelle in a plant cell. It holds wastes and materials, and then it also maintains proper pressure in the cell that provides structure and support. And there you have it. The plant cell. Now you may want to go get your AC1 levels checked.